Hello students, today we discuss about the topic restriction enzyme that comes under the genetics B.VOC.MLT 405. First of all, you have to understand what do you mean by restriction enzyme. Actually, it is a type of a Caesar. You know Caesar, how Caesar work. Only the tailor know how where the cloth to from where we cut the cloths, right? If we cut elsewhere, then the whole cloth will be waste. Like that only. Restriction enzymes are the enzyme which cut the nucleotide base pair. But at a specific point, remember this. At a specific point, restriction enzymes are the enzymes which cut the DNA at DNA at the specific base pair of the nucleotide. For example, eco R1. See here, this is the eco R1, right? Eco R1 cut at a base pair GAATTC. Where GAATTC are present, then only this eco R1 cut this base pair. Clear? So, every restriction enzyme cut at a specific point in between there's in between the recognition sequence or some away from the recognition sequence, but cut at a specific point, not elsewhere. And see, this is a sequence GAATTC, right? And here also GAT. This sequence is known as a palindromic sequence. From here we read and from there we read. These sequences are equal, right? Here we read here from GAATCs, and here also we read same GAATTC. This type of sequence is known as palindromic sequence. From right hand side or left hand sides where you read, the sequence will be same. Okay, understand? So now we start our restriction endonucleases. In restriction endonucleases is also known as a restriction enzyme. Actually, restriction enzyme are known as the restriction endonucleases. In 1962, molecular Caesar discovered in a bacteria. Where? In bacteria. Mainly bacteria are a group of organisms in which these restriction enzymes are present. You you hear about the PCR techniques, right? In PCR, the restriction enzymes are active at a high temperature, right? At a 92 degrees Celsius like temperature. So in that type of restriction enzyme, this type of restriction enzyme are present in a bacteria which survive at a high temperature, and that bacteria is known as thermophilus bacteria, which love the high temperature. We survive that bacteria survive at a high temperature. So coli bacteria have an enzymatic immune system that recognizes and destroys foreign DNA. 3,000 enzymes have been identified. Around 200 have unique properties. Many are purified and available commercially. Actually, restriction enzymes are of two types. One is exonucleases and one is endonucleases. But mainly that enzyme that are used in a molecular biology, that is restriction endonuclease. So restriction endonucleases is mainly known as a restriction enzyme, or restriction enzyme is known as a restriction endonucleases. First of all, what is the history of the restriction enzyme? The first restriction enzyme that were isolated are in 1917, and the first restriction enzyme are hint 2. He he also done the subsequent discovery and characteristics of numerous restriction endonucleases. Already I know that there are 3,000 restriction enzyme are present, right? 3,000 restriction enzyme are now present. But 600 or more than 600 are available commercially and are routinely used for DNA modification and manipulation in the laboratory. There are different types of restriction endonucleases. There are three types of restriction endonucleases: type 1, type 2, and type 3. Type 1 are a bifunctional enzymes. Bifunctional enzyme means they their function are of two types, because they are made up of three subunits like alpha, beta, gamma. They are made up of three subunits, and their recognition site are bipartite and asymmetric. This restriction enzyme are naturally exclusive, and their cleavage site is more than 1000 base pair away from the recognition sites. Means like that we GAATTC in eco R1 recognition site are what? GAATTC, right? Then that only GAATTC eco R1 cut in between this sequence. See, right? G eco R1 cut only at in between that sequence, but type 1 restriction enzyme cut away 1000 base pair away from the recognition sequence. Means GAATTC they presents, and then type 1 restriction enzyme cut away from this type 1 from this recognition sequence like GAATTC. Type 2 separate endonucleases and methylase means they have a separate function, endonucleases and methylase activity. Type 3 have same as type 1 bifunctional enzyme. They act as both endonucleases and methylase activity. Their recognition site are type 2 4 to 6 base pair sequence, often palindromic. Palindromic means like GAATTC from left hand side and right hand side. When you read from left and right, both the sequence will be same. That is known as palindromic. Clear? And in 5 to 7 base pair type 3, but it is asymmetric sequence. They are not palindromic. Type 1 and type 3 both are asymmetric sequence, means when we read from left hand side or when we read from right hand side, so both the sequence will be different, not the same, like GAATTC, not same. So type 2 have a separate reaction, means they have a both endonuclease activity and methylase activity, but they have both their activities are separate, not same at the same time. But in type 1 and type 3, both the methylase and endonuclease activity work at a simultaneously. Type 2 cleavage sites same as or close to recognition sites. Means type 2 restriction enzyme cut at the recognition sequence or close to the recognition sequence. Clear? Means eco R1 example of type 2 is eco R1, because in eco R1 they cut the sequence, they cut the sequence at between the recognition sequence. Type 3 24 to 26 base pair downstream of recognition sequence. So only the type 2 restriction enzymes endonucleases enzyme are the enzyme which cut the which remember the which cut the base pair at the recognition sequence. That's why type 2 enzyme are very important in molecular biology. So now we discussed in detail about type 1, type 2 and type 3. Okay. So type 1 cut 100 base pair after the recognition sequence. Already you see that. Type 2 cut at the recognition sequence. Type 3 cut 20 to 30 base pair after the restriction sites, or they cut at the downstream. Remember this word downstream. The type of enzyme used depend on the desired outcome. which what was their outcome, what you want to observe. According to that, only we select the types of restriction enzyme. So first we cover type 1. Type 1 capable of both restriction and modification activity. What? Both restriction and modification activity. The cofactor that present in type 1 is S-adenosyl methionine. Remember this, the cofactor, adenosyl methionine, ATP, and magnesium ion. These are required for full activity of type 1. If there are one cofactor is missing from this, then the activity of the type 1 restriction enzyme is blocked. And they contain two R subunit, two M subunit, and one S subunit. Clear? They contain two R subunits, two M subunit, and one S subunit. R means restriction for restriction enzyme, M for methylation, and S for specific subunit. For R means restriction means they cut at the specific point. Methylation means they add the methyl group, and S means they are a specific subunit for some specific reaction. Cleave DNA at random length from recognition sites. They cleave the DNA at random length from the recognition sites. So that's why type 1 restriction enzyme are not used too much. Type 2 restriction enzyme. Type 2 restriction enzyme are most commonly available and are used restriction enzymes. Clear? Type 2 restriction enzyme are most commonly available restriction enzyme, and they are mostly used restriction enzyme. They are composed of only one subunit. In type 1, you see that type 1 is made up of three subunit, two R subunit, two M subunit, and one S subunit. But this type 2 composed of only one subunit. Their recognition sites are usually undivided and palindromic and 4 to 8 nucleotide in length. Clear? They recognize and cleave DNA at the same site. What they do? And what they do? They recognize and cleave DNA at the same site. Remember this term. They recognize and cleave DNA at the same site. They do not use ATP for their activity. These are the only enzymes which not use ATP for their activity. They usually require only magnesium ion as a cofactor. Only magnesium, but in type 1, adenosyl methionine, ATP, magnesium ion, this all required as a cofactor. Now type 3. Type 3 restriction enzyme recognized two separate non-palindromic sequences that are inversely oriented. What? They are inversely oriented. They cut DNA about 20 to 30 base pair after the recognition sequence. They cut downstream. Remember this, downstream of the 20 to 30 base pair after the recognition sequence. This enzyme contain more than one subunit like type 1 and require adomethionine and ATP cofactor for their role in DNA methylation and restriction. In this, they not require magnesium ion as a cofactor. They require adenosyl methionine and ATP as cofactor. Now see, restriction endonym for bacteria like this. Example of EcoR1. In first slide already you see that EcoR1. How the name was specific. How this name are specific. See, this first word E. Remember this. This represent the genus. Co represent the species name. R represent the strain, and one represent this number represent the order of discovery. What? Order of discovery, where when they are discovered. Clear? So that's why this is known as EcoR1. Means this genus means E represent is Escherichia, Co represent coli, R represent strain, and one represent order of discovery. Right? That's why their name is Eco R1. According to that only hint 2. Right? So name for bacterial genus, species, strain, and type. These are the these are these are the how we write the restriction endonucleases. Now, recognition sites have symmetric, that is palindromic. What do you mean? Already I told you what is palindromic. Palindromic means from left side we read or right side we read. Both the sentence will be similar. So these are the site of B.VOC.MLT site. In previously we see that the eco R1 site. These are the B.VOC.MLT site. Clear? So B represents the genus, M represents species, H strain, and what order of discovery. So their site was GGATCC. So when we read from here or here, not you think that we read this sentence from same strand from opposite. No, from one strand from right side, then in second strand we read from left side, not in same strand. Remember this thing. Clear? In DNA, in restriction enzyme, palindromic means we read the sequence from left side in one strand, then we read the sequence from right side from the in another strand. Clear? So GGATCC, GGATCC. These are the recognition sites. Enzyme recognize a specific 4 to 8 base pair sequences. What? They recognize specifically 4 to 8 base pair sequences. Some enzyme cut in a staggered fashion and some enzyme cut as a that is sticky ends, and some enzyme cut in a direct fashion, that is known as blunt end. Sticky end means staggered fashion means in see this is the eco R1. They cut the sequence at from this after G and here after G. So when they cut, what happened? This will be like that only, right? This will be like that only, right? Then what happened? Here one base pair will be empty, here one base pair will be empty. So this is look like a sticky end. Means one another another sequence will come and stick with this. This is known as sticky ends. But in this direct fashion means in this suppose that one restriction enzyme cut at this, right, after this G, and from here this cut at this position. So what happened? This cut and hold this and hold this. So what happened here? What happened to here? This both the sequence will be removed. T, G, T, A, C. This will be removed, and from here also this AC and this TG will be removed. So this is known as a blunt end. Means there is no sticky, no extra base pair is present, right? So this is known as a blunt end. See here. See here. This is the sticky end, and when this will be cut at this position, like between GC, then what happened? What happened? Here blunt end will be produced. So when the restriction enzyme cut the sequence, there are two types of end will be produced. That is, one is sticky end and one is blunt end. See. From this diagram, the sticky and blunt end will be clear. See, eco R1 cut at GA from here and from here GA, right? Cut, cut this. Then what happened? This type of end will be produced. So this type of end is known as sticky end. What? This type of end is known as sticky end. But in this, see, HAE3. What happened here? They cut the at this point. Means there is no extra base pair is present. This is known as blunt end. Clear? Clear the difference between sticky ends and blunt end? Clear? Right? So this is known as a sticky end and this is known as the blunt end. Clear? So why don't bacteria destroy their own DNA with their restriction enzyme? This is the question comes in your minds. If bacteria if bacteria contain the restriction enzyme, because all the restriction enzyme, maximum restriction enzymes are isolated from the bacteria, then why not that restriction enzyme destroy their own DNA? Why? Because because bacteria, every each and every DNA having a methylated group, which inactivate the DNA property. That's why restriction enzyme not cut the bacteria itself. Clear? Only because of methylation. Because when methyl group will be present, means means the DNA will be inactive. DNA will be inactive, means they are not active at that time for the restriction enzyme to come and cut. Clear? So see, this is the unmethylated DNA, and this is a site of eco R1. Eco R1 come and cut this sites, come cut at this site and produce the sticky end. If see here, this is also a methylated group. Eco R1 methylase come and cut. If here methyl group are present, if here methyl group are present, then what happen? Eco R1 will not come and cut this sites. Eco R1 will not cleave methylated DNA. Remember this. If DNA are methylated, if DNA are methylated, then DNA will not, sorry, restriction enzyme will not come and cut the DNA. Remember? Remember this? So, for the cutting of the restriction enzyme, unmethylated DNA will be compulsory. Clear? What are the use of restriction enzyme? Restriction enzymes are used in RFLP analysis. RFLP analysis means restriction fragment length polymorphism. DNA sequencing, DNA storage, transformation, large scale analysis. In large scale analysis means gene chip. So first, restriction enzyme used in transformation. How the restriction enzyme are used in transformation? See, this is the human DNA cleaved. First of all, what is the transformation? Transformation means when we want some gene of interest to insert in another organism, that is known as transformation, right? That is known as transformation. Means we add our gene of interest in any organism to see the activity or properties of that gene in that particular organism. This method is known as transformation. This is a human DNA cleave with eco R1. Human DNA cleave with eco R1. Corn DNA cleave with eco R1. Remember one thing also, that when we use the process of transformation, then donor and recipient, both the DNA will be cleaved with same restriction enzyme, not with different restriction enzyme. So human DNA cleave with eco R1 and corn DNA also cleave with eco R1. This is the human DNA, this is the corn DNA. And this DNA having a site for eco R1 and they cut at the site. Okay. Then what happened? They produce sticky end, because eco R1, when eco R1 cut the DNA, then sticky end will be produced. Then they produce sticky end. What happened then? DNA ligase will become and RATP. DNA, what is the function of DNA ligase? The main function of DNA ligase is help in joining the DNA. What? The main function of DNA ligase is in joining the DNA. So the DNA ligase came and they joined this base pair, right? So this, when they join this base pair, then what happen? What type of this DNA will be formed? This is known as recombinant DNA molecule. What happened here? This is known as recombinant DNA molecule, and this is known as transformation, which transform the original human DNA with with corn DNA. Clear? So this is the application of the restriction enzyme they used in transformation techniques. Now, restriction enzyme for RFLP, that is random fragment length polymorphism. What when we want to identify the sequence, a specific sequence or randomly sequence that are changed in human, we use the restriction enzyme. What happened? We select the DNA, then restriction enzyme cut at a specific point, and then we run a gel. When we run the gel, the pattern of the different types of according to the size they separate on the gel, right? DNA is negatively charged from the phosphate backbone. Visualize DNA with iridium bromide fluorescence only when bound to DNA. Clear? This is the DNA chamber, right? This is a gel chamber. Here we load our sample. What happened here? Here we load the sample. This the sample which contain the fragment of the DNA, right? One is the marker and others three are our sample. Okay. When we load the sample, what happened? The DNA molecule is negatively charged, right? So what happened? They move toward the positive charge. Clear? They move toward the positive charge, and you already know that smaller things will move fast as compared to larger. So a smaller fragment will move faster at the bottom, and larger molecule will present top at the top. So when we run the gel, the this DNA molecule will be separated according to their size. The smaller molecule will come at lower position, and the larger DNA molecule will present at top of the position. And according to the marker, this is the first sample already I told you, that is the DNA marker. So from one DNA molecule, from one DNA gel, we this DNA gel act as a marker. According to that, we identify our sample. So this is are the application of the restriction enzyme. These are the actual gel picture. This is the marker, and this is the gel of our sample. These are the gel of our sample. How they identify. Clear? So this all about the restriction enzyme, how the restriction enzyme are work, and what are the application of the DNA enzyme. So next application is in the DNA sequencing. When we sequence the DNA, when we used the sequencing of the DNA, then what happened? We cut the DNA at a particular point, and then we sequence it. So to cut that DNA, for that DNA, restriction enzyme are used, right? What happened? For that only restriction enzyme are used to cut that DNA sample. So restriction enzyme are also used in DNA sequencing, DNA transformation, and DNA storage. Storage means when we prepare the cDNA library, mRNA library, then also DNA storage restriction enzyme are used. Clear this all? So this is all about the restriction enzyme. Clear? Hope hope this video are clear to all. So thank you.